Hello, my name is Gutwein and I'm a PhD student at the Children's Cancer Research Institute in Vienna. And today I will present you my work, which is called Fishing in Uncertainty, which is part of the Unsure workshop at the MICAI conference in 2024. So the term fishing in the title is a play on words on the term FISH, which stands for fluorescent in situ hybridization, which is a crucial method in cancer diagnostics. FISH helps us to identify clinically relevant subtypes in cancers such as breast cancer and neuroblastoma. FISH allows us to visualize the number of copies of specific genes as well as the cell nucleus in individual cells. Here you can see an example of 3 images showing the increasing number of gene copies of the gene marked in green. Currently, these images are evaluated manually in a diagnostic protocol, which is both time-consuming and costly because hundreds, if not thousands, of these images have to be evaluated by more than one expert. FISH presents its unique challenges due to the unclear definition of signal numbers, often caused by signal proximity and blurriness, leading to difficulty in evaluating these uncertain cases. Our focus in this project is on 3 clinically relevant cases of the MICN gene, which is a common mutation in neuroblastoma. Normal cells show 2 red and 2 green signals. Then we have the MICN gain, which shows more than 2 but less than 8 green signals while maintaining 2 red signals. And we have a MICN amplification, which shows more than 8 green signals. This gives us 2 decision boundaries between 2 and 3 green signals and 7 and 8 green signals. Especially around these decision boundaries, the uncertainty in classification is high. To address this, we sought a computational method that can classify cases accurately and estimate classification uncertainty. Due to the scarcity of annotated data, we generated a synthetic data that allows us to densely populate the image space with both clear and ambiguous cases. We developed a tool called Fish Painter, which takes a cell nucleus as input and generates a fish image based on a user-defined configuration. The goal was to train our model on synthetic data and test it on real-world data. For this, we trained a model using a joint loss function that combines a contrastive loss and a cross-entropy loss for classification. The contrastive loss aims to capture intra-class differences, while the cross-entropy loss maximizes the classification accuracy that we are interested in. This approach helped the model to handle complex intra-class variations while maintaining high accuracy. We compared the model's classification accuracy on a synthetic test set with the performance of 10 independent human experts. Our model matched the best annotator and outperformed the average expert by 2%. Next, we calculated the inter-expert agreement for the number of green signals shown in the yellow line. We then compared this inter-expert agreement to our model's aleatoric uncertainty estimates, which we calculated using the normalized cross-entropy over the sigmoid outputs of the network. What we were able to show is that the uncertainty estimates align well with the inter-expert agreement, particularly around the classification boundaries between MICN gain and MICN amplification. As we are mainly interested in the performance on real-world data, we tested our model on a highly curated real-world dataset. As shown here in green, we achieved an overall accuracy over the whole dataset of approximately 86%, which increases when discarding uncertain cases, conditioning on the uncertainty. It increases to 95% for the most 75% certain cases, and even going up to 99% for the 30% most certain cases. We also compared our approach to existing methods like Big Fish and the trained retina model from a different publication, which don't allow for uncertainty estimation. Additionally, we tested training alternative approaches that used only cross-entropy or contrastive learning for training, or contrastive pre-training and fine-tuning using cross-entropy. We can show that our model outperforms all of these methods in terms of classification accuracy as shown on the screen and estimated calibration error as clarified in the publication. As the next steps, We plan to evaluate this approach with the diagnostic team to assess its impact on time and personnel savings in a clinical setting, as well as move forward to come up with guidelines on how such tools could be used in everyday diagnostics, utilizing the uncertainty the model yields us. And with this, I want to end my talk. And if you have any questions, feel free to reach out to me via my mail showing on the screen or following the QR code on the screen, which leads you to the associated GitHub repository of this publication. Thank you.